Here we are at EULAR 2025, day 3. I'm Dr. Valentina Puccino. I'm assistant professor and immunorheumatologist at University of Pisa, Italy, and I'm delighted to be joined here by Professor Constantino Pitzalis. He is director of the Center of Experimental Medicine and Rheumatology at the Queen Mary University of London and professor of rheumatology at Humanitas University in Milan. Congratulations, Cos, for all the work you have done in the biomarker-driven trial field. And I have so many questions for you. And first of all, why do we need these biomarker-driven trials, and where are we standing now?
Thank you, Valentina. Conventional clinical trials have really transformed the life of many patients with rheumatoid arthritis. However, we know that 40% of patients currently do not respond to individual biologics, and 10 to 20% are refractory to all medications that we have. So we need new clinical trials, biomarker-driven and patient-centric, so that we can tailor the medications to the individual patient.
Thank you. And do we also have biomarkers in the peripheral blood that can inform such trials?
So research in the last 20 years has been unfortunate in terms of determining biomarkers in the peripheral blood to predict treatment response. We have good biomarkers in the peripheral blood of disease activity. So we know when the patient has a high CRP, for example, that patient has bad disease. However, we don't have biomarkers in the peripheral blood of treatment response. So what's happening is therefore that we give these drugs, which are targeted therapies, to patients not knowing exactly whether the patient is going to respond or not respond. Over 10 years ago, we developed an ultrasound-guided minimal invasive biopsy, which enable us to sample the joint where the disease is mainly affecting the patient and develop biomarkers and signatures that can predict treatment response. So that's why we're now designing new clinical trials. They are biopsy-driven. In other words, the molecular profile of the patient will inform which drug we are going to give to the patient.
And regarding this biopsy-driven trial that actually you are proposing, is the procedure safe? Do you see a good compliance from patients?
So the procedure is very safe. Over 15,000 ultrasound-guided biopsy have been performed with very little side effects, minimal side effects, and minimal inconvenience to the patients. So the patients tolerate the procedure well. It takes approximately 20 minutes. It's done under local anesthesia, and so the patient doesn't feel too much discomfort. There are some cases, of course, in which, like any minimal invasive procedure, this can create problems for the patients, but these have been truly minimal, and the patients haven't had any significant side effects.
Very insightful. And now we are at the last question. What do you think will be the future of this biopsy-driven trial? What's the benefit for patients?
So this is very much work in progress. We have a number of clinical trials who are biopsy-driven now, but we don't know the outcome yet of these trials. What we have the results of is previous biopsy-driven trials, which we call a biomarker discovery, which have really taught us an awful lot regarding the pathogenesis of the disease. We understand that clinically indistinguishable patients have a different histopathological and molecular profile, and therefore this informs what is the potential treatment for these patients. Currently, we are using new biomarkers that we discovered in these previous trials to inform the patient's drug that they are going to receive. The results of the trials will be available in the next year or so, and then we'll be able to know whether the synovial biopsy really has clinical utility and, with a molecular profile of the patient, really inform which drug to give to the patient before we give the drug, not after.
Thank you very much. That's very exciting. And thank you for joining me for this interview. And thank you, everyone, for listening and enjoy the rest of the conference. Thank you very much.
Thank you. Thank you.